And this is where we left off the last time. There's something I want to clarify, and that's I think I said reticulocytes are released into the bloodstream. They actually circulate in the bloodstream for about 24 hours before they become mature red blood cells. And there are still some ribosomes, or some RNA anyway, that's filling the between the normal blasts and the reticulocytes. There's still some space for more hemoglobin by the time they're reticulocytes, so they're still creating hemoglobin, but they don't really know why they circulate for 24 hours before they become red blood cells. But the thought is they're basically learning how to do their job, and they're also learning if in fact they are made properly, meaning they're made to be able to carry oxygen. If they can't carry oxygen, then the reticulocytes would be eliminated by the liver. But anyway, going back to blood types, as I said, there are proteins embedded in the membrane, and they're actually integral proteins that, and many proteins embedded in the membrane, not just the ones for blood types. But the ones that tend to be most problematic, meaning cause transfusion problems, is the A, the B, and then the D. The D we recognize as the Rh factor based on the monkeys that were used in experimenting and trying to find out what the Rh factor was. And what those surface proteins that are coded by our genetics is what gives us our blood type. So if you have A surface proteins, you have type A. If you have A surface proteins plus D, which is the Rh factor, you have A plus blood. If you have A and B type proteins on your surface of your red blood cell, you have AB. If you have the AB plus the Rh factor, you have APB plus. And then on with regard to if you have none of the surface proteins, then you have O. No surface proteins at all, you have O negative. If you have none of the A or B surface proteins, you have O, but you do have the Rh factor, then O positive. And actually you can see all of this in this image, and you have a great image in your book, plus we talk about this in lab, but the antigen for A is the little blue dot with the line. The antigen for B is the green arrow with the line, and the antigen for the D or the Rh factor is the kind of brownie. And then as you look through all of them, you can see on the AB positive it has all of those antigens on them. And then look all the way through, and then you have antibodies, and I have an antibody image coming up next. But just a couple of concepts. AB positive is considered the universal recipient, meaning it will pretty much take any kind of blood, and then O negative is the universal donor. And then blood types, as I said, they're based on there are actually three antigens on the membrane that are most problematic. And then with that you have type A, type B, type AB, and the only thing that this particular slide is bringing into the mix is this antibody that's circulating in the plasma. So with type A blood, which you have the A antigen on the membrane, you're going to have the anti-B antibody. So the anti doesn't like B. It's type A, doesn't like B, anti-B. With type B, you're going to have B on the surface, and it's going to have the anti-A. If it's type AB on its surface, there's not going to be any antibodies for that. And neither A nor B, which you have type O, you're going to have anti-A and anti-B. And furthermore, the same holds for the Rh factor. You're going to have the factor. If you have the factor, then there won't be any antibodies. If you don't have the factor, there will be antibodies. The antigen is defined as a protein on the surface of the red blood cell, and antibody are proteins made by lymphocytes, white blood cells, in the plasma that are made in response to the presence of the antigen. So it's an immune response to a foreign invader in our body. If we have if a blood type is introduced that is not compatible with our own. That incompatibility makes this factor of agglutination. So it's clumping. It's not normal clotting. It's clumping of the red blood cells to the point where it can cause a problem. You can have a clot in the heart, a clot in the brain, or a clot in the respiratory system. Any of them, unfortunately, could lead to death. And here's what you will do in lab, or you did in lab, whichever you are when you're listening to this. What you're going to do is you're going to test your own blood, and you're going to put a drop of blood in 2 wells, actually 3 wells, because we're going to do A, B, and then the RH factor. And we're going to look for agglutination. So at the very top, we're looking at the images. What you have is type AB blood, contains antigens both A and B, and will agglutinate with both of the serum. So the agglutination is a test for the fact that you have that protein on the surface of the red blood cell. So this is a chemical that was invented and created to try to understand what type blood, in a very, very simple, very inexpensive way, what type blood the person might have. And so if you have this agglutination, that means you have that antigen on the surface of your red blood cell. The first one, you have both antigens, so that's type AB. The second one, you just have A antigen, nothing reacting on the B, so you have type A. Second one, nothing reacting on the A but on the B. And the last one, nothing reacting on either of them. So you have type O. Now with D, you either have it or you don't have it. So it's either positive or negative. So that's why we usually have 3 wells for whether you're type A, type B, or type AB or O. And then there's a whole separate well to figure out whether you have the positive or the negative. And this is simple blood typing. Blood gets typed for transfusion, looking for compatibility. If in fact there is no compatibility, then we got to move on and find somebody else's blood. With regard to this RH factor, it does become a problem in pregnancy because although the bloods don't mix between the mother and the fetus, antibodies mix between the mother and the fetus. And with regard to RH factor, about 85% and going up in our population is RH positive. Only about 15% and going down is RH negative. And here you have, and I have another slide coming up on the next one too. With the first baby, if the mother is RH negative and the first baby is RH positive, what's going to happen is you're going to have antibodies made by the mother after the course of that baby. So you're going to have antibodies made because the mother's blood is picking up a foreign substance, foreign blood substance, which is actually opposing its type. So antibodies will be made between the pregnancies, and then what will happen is the second pregnancy from an RH negative mother, RH positive fetus, the mother will actually attack it. So the mother's blood will attack the baby's blood because it's not compatible with the second RH positive fetus. And then just to summarize, RH positive mother with an RH negative baby, no problem. because the baby doesn't make antibodies, so it's not going to go the other direction. Rh negative mother with an Rh positive baby, particularly the second one, there's a problem. There is a solution, and that's RhoGAM, used in about 28 weeks. And frankly, anymore, pretty much they'll use RhoGAM. If the Rh negative mother and Rh father is negative, the father and mother both negative, because the only thing that it's double recessive, they're definitely going to have an Rh negative baby, so no problem, and Rh negative mother, Rh negative baby, no problem. So it's just and understand that most of us are Rh positive, so it's just that second Rh positive baby with an Rh negative mother that becomes problematic, and there is something that can be done. Then moving on to white blood cells, it's the next portion of the formed elements with white blood cells, and we divide them up into granulocytes and agranulocytes. If you remember from, well, maybe you didn't take biology, and maybe you haven't taken micro, but this whole idea of Gram staining is that you have particles in the white blood cells that will either pick up violet or will pick up the orangey color, and they'll either pick up the stain. That's what we call the granulocytes. They pick up, they have little particles in the cells that pick up the stain. You can also see them if they're not stained, but you have to have a pretty powerful microscope. And then you have white blood cells that don't have granules. The agranulocytes are lymphocytes and monocytes. So no granules. If you look all the way to the right at the top, a lymphocyte, you can see it's pretty much all just violet. And then monocyte, that nucleus is picking up a little bit, but we're talking about basically in the cytoplasm. But if you look at the fills, neutrophil, which is the most common white blood cell in the lower left, and the interesting thing about a neutrophil is it many times has a multi-shaped nucleus. It looks like it has actually 4 nuclei there. We call them polymorphs because the one nucleus will have multiple different segments to it. We sometimes call them bands. And also neutrophils. So neutrophils, most common white blood cell by far is a neutrophil. It has several different names. Basophil in the top left, that one tends to stain very dark purple in its grains. There's a difference between the lymphocyte and the basocyte in its grains. You can see that that is kind of a granular stain there in that basophil. And the eosinophil, not a very good picture of it, but it stains kind of rosy pink, purpley. You'll see that in lab. Hopefully you get a look at blood cells in lab, if not in the microscope, at least on a image. White blood cells have unique movement. It's one of the red blood cells never leave the circulation, but white cells do. They can migrate out of the bloodstream. They can migrate out of the bloodstream and into the tissues, particularly if in fact there is a need for them in a site of inflammation. We call this diapedesis. Hopefully I pronounce it right. I think I'm pretty close on the mark with that. And you can see that in the image at the bottom that the white blood cell is actually squeezing between junctions between cells. So it actually squeezes between. It comes up to the side of the vessel and squeezes between cells. We'll pick this up on the next one.